Hey there, science fans. Today, we're diving into the fascinating world of amino acids. These tiny molecules are the building blocks of proteins which are essential for life as we know it. Without amino acids, we wouldn't have muscles, enzymes, or even hair. Think of amino acids like Lego blocks. Just like how Legos can be combined in countless ways to build different structures, amino acids link together to form a vast array of proteins. Each protein has a unique shape and function, determined by the specific sequence of amino acids it contains. From the food we eat to the cells in our bodies, amino acids are everywhere. They play vital roles in countless biological processes, making them crucial for our health and well-being. So, buckle up as we explore the amazing world of amino acids and uncover the secrets behind their structure and properties. Let's get down to the basics and examine the structure of an amino acid. Picture a central carbon atom, which we call the alpha carbon. This alpha carbon is like the central hub of our amino acid molecule. Attached to this alpha carbon are four important groups. First, we have an amino group, NH2, which contains nitrogen and acts as a base. Second, there's a carboxyl group, CO, which acts as an acid. Third, a single hydrogen atom, H, is bonded to the alpha carbon. And finally, we have the mysterious fourth group attached to the alpha carbon, the side chain, represented by the letter R. This side chain is what makes each amino acid unique. It's like giving each Lego block a special attachment that changes its properties. Now let's talk more about that alpha carbon and the incredible diversity of side chains. The alpha carbon is at the heart of it all, acting as the central attachment point for the amino group, carboxyl group, hydrogen atom, and the all-important side chain. Each of the 20 common amino acids has a different side chain, which gives it unique chemical properties. Some side chains are small and simple, like the hydrogen atom in glycine. Others are large and complex, containing rings or chains of atoms. These side chains influence how amino acids interact with each other and with water. Some side chains are hydrophobic, meaning they repel water, while others are hydrophilic and love to mingle with water molecules. These interactions are crucial for determining the three-dimensional structure and function of proteins. Chapter 4. Proline, exception to the general structure. Now let's explore an exception to the general amino acid structure, proline. While most amino acids adhere to the basic structure we've discussed, proline stands out with its unique configuration. Its side chain forms a ring that includes the alpha carbon and the amino group. This distinctive structure makes proline more rigid than other amino acids. Think of it as a more inflexible Lego block. This rigidity influences how proline fits into protein structures and can introduce bends or kinks in the protein chain. Proline's unique structure underscores the importance of variation in amino acid structure. These variations allow proteins to fold into complex shapes and perform a wide range of functions. Section 5. Amino acids, amphoteric nature. One of the fascinating things about amino acids is their ability to act as both acids and bases. This amphoteric nature, known as amphoteric behavior, is due to the presence of both the amino group, basic, and the carboxyl group, acidic, within the same molecule. Depending on the pH of the surrounding environment, an amino acid can donate a proton, acting as an acid, or accept a proton, acting as a base. Imagine an amino acid as a quick change artist, switching between acidic and basic forms depending on the situation. This amphoteric nature is essential for many biological processes, including enzyme activity and protein folding. It allows amino acids to interact with each other and with their environment in complex and dynamic ways. Section 6. The isoelectric point, finding balance. Now, let's talk about the isoelectric point, or pI, of an amino acid. This is the pH at which the amino acid exists as a Zwitterion, a neutral molecule with both positive and negative charges. It's like finding the perfect balance point for our quick change artist. At its isoelectric point, the amino acid has no net charge and is least soluble in water. This property is important for protein purification and separation techniques. By adjusting the pH of a solution, scientists can manipulate the charge of amino acids and separate them based on their isoelectric points. Zwitterions are fascinating molecules with unique properties. They highlight the dynamic nature of amino acids and their ability to adapt to different environments. Section 7, amino acids, tiny but mighty, and there you have it, folks. Amino acids may be small, but they are mighty molecules that play essential roles in all living organisms. From forming the building blocks of proteins to exhibiting unique chemical properties, amino acids are truly fascinating. We've explored their basic structure, the importance of side chains, the rebellious nature of proline, their amphoteric behavior, and the concept of the isoelectric point. And this is just the tip of the iceberg. There's so much more to learn about these incredible molecules and the vital roles they play in our bodies and the world around us. So keep exploring, keep asking questions, and stay curious about the amazing world of science. For more insights into the life sciences, subscribe to our channel.